Okay, can you see my screen?
Yep,
All right,
perfect.
great. So, let's see. My thesis was on the electrophysiologic response to classical music in instrumentalists, vocalists, and non-musicians. So the objectives of this thesis was to be an exploratory case study with the following questions in mind. So to investigate whether individuals with different musical backgrounds have greater electrical activity in different areas of the brain when listening to classical music, and if so, which areas. To identify whether musicians listening to music within their expertise have a greater electrical response than when listening to less familiar music, and if so, which areas of the brain are most active. And to compare and contrast spectral mapping and the parametric orchestral sonification of EEG in real time, or the poser method, of their ability to present data while still being aesthetically pleasing. So the past research that informed this study includes a longitudinal study that's revealed structural differences in brain development between individuals who have received musical training and those who have not. And in this study, they found that after only 15 months of musical training, individuals that underwent that training had a greater relative voxel activation in their motor cortex, right precentral gyrus, and Heschel's gyrus. So I approach this as looking at whether these structural differences cause functional difference as well. Another study that informed mine was a study that found that musicians more actively process changes in music in their left hemisphere when compared to non-musicians in a study. And these studies examined EEG responses in non-musicians and musicians to major changes in music, such as tempo, melody, key changes, and dissonant or seemingly wrong notes being played. So these are all considered event-related potentials, or ERPs, which means that the researchers were looking at the change directly after receiving the specific stimulus. So I'm interested in seeing whether continuous stimuli have similar responses. So then the third study and idea that informed mine was a study surrounding the differences in discrimination and production of musical pitches in instrumentalists, vocalists, and non-musicians, and it found that the vocalists were more consistent in their production than instrumentalists, but both were more accurate than non-musicians. So this has led to the hypothesis that vocalists are more consistent than instrumentalists due to their expertise and experience in this skill. So I'm interested in seeing whether these differences in expertise translate to the electrophysiological response. So in order to do this, I collected data using EEG, which is a method of recording and measuring electrical activity within the cerebral cortex via electrodes placed externally on the head. The electrical activity that is recorded is created by neurochemical signaling within the brain, specifically the NMDA receptors becoming permeable to calcium ions, which creates an output with the electrical activity of the electrode over time. So EEG is measured using, the EEG measures the neural oscillations, which can be broken into frequency domains. So gamma waves are the highest, or are high frequency, and it implies arousal and are generally involved with learning, memory, and information processing. Beta waves are the next highest frequency and are involved in conscious thought. Alpha waves are produced during general relaxed calming states, and then theta are slower during a resting state, and delta during deep sleep. So in this, in my study, I'm most interested in looking at increased percentages of beta and gamma waves. So I was also interested in looking at sonification a bit. I didn't quite get to this part, but this is what my goal was. So sonification is a way to present data in an auditory context. So sonification is thought to be an effective method for uncovering structures and patterns within data because of the capacity for our auditory system to process and understand multiple auditory streams at once, whereas the human visual system must process multiple objects through serial processing. So sonification can be done using a variety of methods. However, it's critical that the method that's being used is able to be aesthetically pleasing, but not transform the data to be unrecognizable. So therefore, different methods of sonification are used for different purposes. So in this study, I was interested in looking at two different types, spectral mapping sonification and the modified parametric orchestral sonification of EEG in real time, or the poser method. So spectral mapping sonification involves each channel, in this case the electrodes, being sonified on its own with increased frequency leading to increased pitch. And this allows for the electrodes to be superimposed on top of one another so that they can be easily compared. And then the poser method assigns frequency bands to different musical instrument digital interfaces, or MIDI channels. So they each have a different timbre, and the pitches are then controlled by the percentage of that frequency band in the area of interest. So this method allows for many channels to be heard at once, and it was originally created to be used in real time, but it's easily modified to be done post data analysis. So the methods for my study were, I had 3 participants total, 1 in each category, and they were categorized as either an instrumentalist with 5 or more years of instrumental training and they've played in the past year, a vocalist with 5 or more years of vocal training who have sung in the past year, or a non-musician who has less than a year of training and does not currently play an instrument or sing. So they were given a brief survey to gain information on their musical background, and then an EEG was recorded while they listened to 6 one-minute excerpts of unfamiliar classical music with 15 seconds of silence between each example. And the excerpts were drawn from a variety of composers with an equal number of instrumental and vocal pieces. So these are the musical stimuli that I used. The pieces chosen were a variety of major, minor, fast, and slow tempo, with 3 primarily vocal and 3 instrumental pieces. And all pieces were written during the same time period, the 18th, 19th century, and all composed within the rules of the common practice, which is the tonal system and rules that we use in Western classical music. Essentially, it's our corresponds with our perception of consonants and overall pleasant music. So all the pieces were also chosen to have little or no rhythmic deviance or tempo changes due to the fact that we know that ERPs do cause a response, and I'm trying to look at continuous stimuli. So the 6 excerpts that were chosen each represent a category of classical music in order to assure that the participants in the instrumentalist and vocalist categories were played at least one piece that aligns with their typical practice and performance. And all pieces that were chosen are lesser known, but composed by well-known composers. So this is so that the music has similarities to what the musicians often play or sing without being immediately recognizable. So this is all done with the help of Professor Platoff in the music department to ensure that the pieces that were chosen are not very well known. So for my analysis, I used EEG Lab, which is a MATLAB plugin, and it was used to analyze the data. First, noisy channels were eliminated, then an independent component analysis was run, which essentially, ICA is a statistical signal processing technique with the goal to express a set of random variables as linear combinations of statistically significant component variables. So it's creating meaningful group signals, essentially. And on the left, you see one of my, after ICA analysis for my second subject, it creates less channels to work with and groups them meaningfully. Then components were examined and rejected based on their validity, including muscle movements and eye movements where components were rejected, and time frequency plots and fast Fourier transforms, or FFTs, for channels and components of interest were created. So going into my results, so for my non-musician, it was a 22-year-old right-handed female with no vocal or instrumental experience who currently listens to classical music about 2 hours a week while engaging in other activities. So on the left, we see a graph that is a time frequency of the first independent component. So here we see a slight increase in the beta gamma waves as we see the frequency on the y-axis during the female opera piece. And then there's a greater centralized activity around the frontal electrode, specifically F1, with the most spectral density between 6 and 10 hertz, which is the theta and alpha waves. So this is seen on the figure on the right, the power spectral density figure. And so the implications of this is that there still is a response to some stimuli more than others in a non-musician. So my instrumentalist was a 22-year-old right-handed female with 12 years of instrumental experience in piano, clarinet, and saxophone, with 2 years of choral experience, and is currently singing more than they're playing. So they've studied music history for 1 year and music theory for 4 years, and they currently listen to classical music for about 4 hours per week. So again, we see on this time frequency plot an increased gamma beta waves that fall on the mixed choir piece, the female vocal solo, a bit on the instrumental symphony, and on the instrumental string quartet. So this increased prevalence of beta gamma waves indicates a greater response to these stimuli during these times. So this is a bit counterintuitive to what I originally thought. I thought that the instrumentalist would have a greater response to instrumental music. However, this individual is currently singing more than they're playing, so that could have some implications in these findings. We also see on the right that the spectral density is similar to the non-musician, all kind of focused around the frontal electrodes. So my results for my vocalist. My vocalist was a 21-year-old right-handed female with no history of instrumental training. with 14 years of choir and 10 years of voice lessons with a focus on opera. She currently sings approximately 12 to 14 hours a week and has studied music history for 3 years and music theory for 2. And she currently listens to classical music approximately 6 to 8 hours per week. So here where we see the most beta gamma waves increase during the female opera piece, which is the female solo in the middle. And there is also some increase in prevalence of these gamma waves in the instrumental, symphonic, and the string quartet pieces. Again, looking on the right in the spectral density, we see everything is more localized around channel 17 towards the frontal and a bit towards the left hemisphere. So for the discussion, it was found that individuals of varying musical backgrounds seem to have the greatest electrophysiological response in the same brain areas. There's no major difference between where these, where the response was. It was all kind of located around the F5, F3, F1, FC5, FC3, and FC1 electrodes. So while these results can't be tested for any significance due to the super small sample size, and the results collected can still lead to conclusions about the individuals. So it was the non-musician had the increased response to the female vocal piece a bit with little response to anything else. The instrumentalist had the increased response to the mixed choral piece, orchestral piece, female vocal piece, and string quartet. And the vocalist had the largest response during the female vocal piece, with some during the mixed choir, orchestral, and string quartet, which aligns with their musical expertise. So the increased beta and gamma waves lasted for 20 to 40 seconds, not the full duration of the musical stimuli. So this could indicate that that there was some kind of event during this time that caused them to respond, not the actual stimuli, which was seen in past ERP studies. So, and that is something to look for in the future. Also, at about 170 seconds, everyone had a response, with the vocalist being the strongest response, then instrumentalist, then non-musician. This was during the female solo vocal piece. So this could have been created by some kind of ERP event. And so when going back and listening, I'm just going to play, I don't know if you can hear this, but I'll just play the brief section which this response seems to happen. So there is a slight change in the mood of the piece, but I didn't think that it would be enough to create an ERP. So, but that is interesting and something to potentially look at and to keep in mind. Oops. So some takeaways from this study are just that musical experience and background did affect the electrophysiological response to musical stimuli, and that all individuals had the greatest electrophysiological response in similar brain areas, and that in the case of the vocalist, the stimuli that they responded to most strongly was the stimuli that did align best with their musical expertise and experience. All right, so I would like to thank Professor Raskin, Professor Lloyd, and Professor Platoff for his guidance in choosing musical stimuli, and I would also like to thank Ross and Elaine for their help in data collection. Thanks.
Yay!
That was awesome, Brielle.
Any questions for Brielle?
Can I ask a?
Yeah.
Yeah, so thanks for the talk, it was great. I was curious, the type of response, since they all responded to the female vocal music, is there a similar response to non-musical speech that you'd expect from the subject? So,
I'm not really sure. That would be interesting to see. I know that there has been some research done on like listening to or processing sentences specifically, and like processing language. And there has been found to be a difference in musicians and non-musicians. I'm not sure if there's a difference in vocalists and instrumentalists, but there does seem to be a connection between language and musical experience.
Thanks.
Yeah.
Any other questions? Students, you can type in the chat box if you're having trouble with internet. All right, thank you, Brielle. So
Thank
while Brielle was talking, it occurred to me that I didn't actually give an introduction, but listening to her talk about this amazing study that I personally know was a huge effort throughout the year, it reminded me that I didn't do my usual introduction, which is to acknowledge that this day every year, whether we're in person or not, is the culmination of a tremendous amount of work on the part of these students. All of them for at least a full year. Many of them started work in labs even before their senior year and have been working on these projects for a very, very long time. And it's always so impressive and so gratifying to see the level of work that they're able to complete as undergraduates. And I know that when all of our students go to professional conferences, people are always shocked to learn that they're undergraduates doing this level of research work. So I just want to acknowledge all of our presenters today and all of the faculty who supervise them. Sorry I didn't do that before yours, Brielle, but yours was so good that it reminded me that I needed to do that. I also want to make sure that everyone knows that I am recording. If you object to being recorded, I can splice you out later, but I just thought people could, because we're in this format, share it with family members or students who couldn't make it or people in odd time zones in case they wanted to. All right, so I think up next we have Maddie. Maddie, are you ready?
Yep.
All right, and Maddie's been working in Professor Helt's lab.
Sorry, real quick, Professor Raskin. Would it be possible to gain access to the recording? Because I know since there was a time limit, the presentation was kind of fast, so I wasn't able to catch all the major points.
Sorry, I was muted. Yes, absolutely. That's great. As long as the presenters are okay with it, I'm happy to make it available to anybody.
All right, thank you.
Okay, so I'm unmuted, right?
Yeah, you're good.
Okay. So for my thesis, I worked with Professor Helt in analyzing a database that we got from the Connecticut Children's Medical Center in order to see if we could find sex differences in restricted and repetitive behaviors and interests in children with autism spectrum disorder. So just a brief background on autism spectrum disorder, ASD is a neurodevelopmental disorder characterized by deficits in the 3 domains of social communication, social interaction, and repetitive behaviors. And it's important to note that each of these domains can vary in severity and typology. So for our study, we focused on the restricted and repetitive behaviors and interest domain, as it is the least studied of the 3 domains. Restricted and repetitive behaviors and interests, or RRBIs, are behaviors characterized by high frequency, repetition, and desire for sameness in the environment. And the DSM-5 and the ICD-10 criteria divide RRBIs into 4 categories. The first category is stereomotor, which is, you can see in the top right of the boy flapping his hands. The second is rituals in the top left photo with the little boy playing with cars for no particular reason. The third category is restricted interests in the bottom left photo of the boy playing with only cars. And then the bottom right photo is of a little boy holding his ears, representing hypo or hypersensitive sensory. So for our study, we specifically investigated sex differences in RRBIs. Previous studies of sex differences on RRBI severity and typology have yielded mixed results. So some studies have found no differences in RRBIs between males and females, whereas other studies have found that females show significantly less RRBIs than males do. So I organized these previous studies into a chart laying out the main information to try to find specific patterns. And these 3 studies here found similar findings with no significant differences in RRBIs between males and females in preschoolers. And this one study in the middle by Harrop and colleagues stood out as, although the data was not significant, it suggests a trend that as kids with ASD get older, RRBI sex differences become more apparent and females show lesser RRBIs than males do. In other words, females can do this thing called camouflaging, where they can hide or camouflage their RRBIs because as they age, they participate in more behaviors that are seen in typically developing females. Whereas the difference between behaviors in ASD males and typically developing males is much more noticeable. So because of this camouflaging, there is suspicion that these results could be inaccurate. So here are some studies that did find significant differences in RRBIs between males and females. And we see here that these studies investigate older participants in elementary school, relating back to this idea that older females may be better at camouflaging at older ages. However, these studies here are limited by small sample sizes, and they're also either using only a clinician report or only a caregiver report, which has these two methodologies have shown disagreements in the past. Also, diagnostic measures for ASD are often biased and created from the male ASD phenotype, so leading to biases and inaccuracies in diagnoses. And these factors may be the reason that the male to female ratio in ASD is so high, and thus also for this inconsistent female phenotype in ASD. So not only have studies shown differences in RRBI rates and severity, but they also have shown distinct patterns. So for examples, in one study, females tend to show more compulsive sameness, restricted, and self-injurious behaviors. And there was also reported a greater overlap of ASD and obsessive compulsive symptoms, such as hoarding and hair pulling in girls. So this literature, based off of this literature, we proposed two questions. The first regarded severity of RRBIs. So do females show fewer RRBIs than males do? And we wanted to use the total scores from the clinician ADOS report and the PDDBI parent report, but due to the global crisis, we were only able to receive some of this data from the CCMC. So we used the PDDBI measures for this particular question. And we predicted that males would have higher overall summary scores than females, especially in older children, as females may become more adept at camouflaging as they age, and ASD diagnostic measures may be biased. So our second question is in regard to the patterns of RRBIs. And we asked, do females and males show different patterns of RRBIs as reflected in distinct types and subcategories of RRBIs as reported on the PDDBI parent report and the DSM-5 clinician report? And again, here we did not have the PDDBI numerical data for this, so we used the DSM-5 clinician report only here. And we predicted that females would endorse more RRBIs associated with cognitive inflexibility, potentially reflecting habit formation. So for our method, our participants, the data was collected previously as a part of an ongoing genetic study at the CCMC, and we compared the 90 females in the database to the 168 males in the database. But our future goal, when we do receive all the numerical data and the IQs of these individuals, we want to ideally match 90 females with 90 males, 1-to-1 by age and IQ. For measures, we wanted to look at multiple reports and the RRB domain scores, but again, we were limited with only part of the database. So for the severity and rate of RRBIs, we used the PDDBI total T-score data to analyze. And for the typology and patterns hypothesis, we use the DSM-5 symptom checklist for analysis. So for analyzing the severity and rate of RRBIs, we used a MANOVA to test for gender differences on variables related to RRBI severity. So for this, we used the PDDBI total T-score. And then for analyzing the patterns of RRBIs, we used a binary logistic regression because the data that we received from the DSM-5 checklist was categorical. So either yes, the individual showed these behaviors, or no, the participant did not show the behaviors. But once we receive all the numerical data, we plan on doing a profile analysis of the 180 matched participants. So the first two slides, which you'll see next to my results, are the logistic regression data based on the DSM-5 criteria, which is completed by a professional in a clinical setting. So the first three criteria here are more so representative of deficits in the social and communication domains of ASD. So the social, emotional, nonverbal, communicative, and relationships, whereas the bottom four are more representative of the RRBI domain of ASD. So like I said before, the stereomotor, the sameness in routines, restricted interests, and sensory behaviors. And we looked at all 7 of these to see if there were similar findings to previous research that found more sex differences in the RRBI domain than in other domains of ASD. So here shows the social and communication criteria of the DSM-5 checklist, which includes, again, the social, emotional, nonverbal, communicative, and relationships data. And the data is presented as the percentage of individuals who showed deficits that clinicians endorsed in these areas. And as you can see, there are very minimal differences between males and females in each of these criteria, as the greatest difference is about 2%. So this shows that clinicians endorse similarly for males and females in the social domain. The percentages are a bit different here for the repetitive behaviors. And I just want to point out that the darker pink represents the percentage of females and the lighter pink represents percentage of males. And although stereo, motor, and sensory behaviors are about equally as likely to have a clinician endorse them for a male or female, there are drastic sex differences in endorsing the rituals and the intense interests. So the data indicate that females are more likely to show obsessive rituals and males are more likely to show intense interests. Since clinician reports such as this, the DSM-5 checklist, observe children over a shorter period of time, it's important to also look at a parent report in which a parent obviously observes their child over a longer span of time. So that's why we looked at the PDDBI total T-scores here. So we looked at the mean severity scores in each RBI symptom domain as endorsed by caregivers on the PDDBI caregiver report, where they can rate severity 1 through 5, with 1 being not severe and 5 being extremely severe. And we found similar trends here to the DSM-5 checklist, where caregivers reported rituals of their daughters to be much more severe than caregivers reporting their sons, and caregivers reporting intense interests in their sons to be more severe than caregivers reporting their daughters. So you can see down here, based on the p-values, that we have significant differences in the rituals and interests sections, whereas there's no significant differences between males and females. And again, the stereomotor and the sensory. But this slide and the last slide just showed there were agreement between the clinician and the caregiver reports. So our overall conclusion, the results were consistent with previous results that rituals may be more representative of a female profile and of greater intense interests in males. So greater hoarding behaviors, which are characteristics of OCD, and thus greater overlaps with OCD, as well as greater self-injurious behaviors in prior studies, are in line with our findings. The significantly less severe interests in females suggests that they may be better at hiding their intense interests, as they may be missed due to how normal the interest may seem, as they may only be abnormal in their intensity. For example, new research has found that many females with eating disorders have ASD traits, showing how females and males may show different ASD phenotypes, and that female phenotypes may be seen as more typical. And all of this indicates that a need for improvement in diagnostic measures for females being addressed or being assessed for ASD, as again, these measures may be biased towards recognizing more male symptoms than female symptoms. And lastly, I'd just like to thank Professor Helt for her guidance throughout this whole process, despite the circumstances, as well as the thesis committee and the neuroscience department over the years. And lastly, to Professor Eichste at UConn and the CCMC collaborators who allowed us to have access to this database, and without them this wouldn't be possible. So thank you.
Yay.
Excellent. Any questions for Maddie? All right.
Great work, Maddie.
Thank you.
Okay. You have a question from Alicia in the chat saying, are there any diagnostic measures that can address camouflaging?
There is a new measure that is specifically about camouflaging that we're using in a different study. So it's pretty exciting, actually.
Cool. Good question, Alicia.
I'm sorry if I missed this, but is there an equivalent for like why they don't recognize the rituals as often in the males? Do you know what I mean? So you saw the disparity both directions.
Yeah. So there was one study that was about teachers observing from a distance, and typically developing females, they tend to be more social, and they found that girls with ASD tended to stay in closer with those groups. So from a distance, they didn't look as if they were doing anything out of the ordinary. Whereas with the males, they are typically developing males, they usually, participate in games with rules. So it's a lot easier to see those males kind of stray and wander away from those structured games.
I have a quick question. Did you mention, Maddie, the age of your children that were participating?
Yeah, so that's another thing. So we were very limited with the data that we had. So we weren't even given the ages. We know that they are children and adolescents. So anywhere from, I think, 3 to 18 years old. But for the data that we have now, they did not give us specific ages. But we're supposed to be getting that in the next few weeks and then publishing hopefully something significant over the summer.
Okay.
These are all medical doctors who entered the data, so they thought it was ready for us psychologists to analyze and presented it as such. And then Maddie and I got it in March and we're like, oh, there's stuff missing, like date of birth that we need. And then the crisis happened and it's been really hard to get anyone to enter that additional data, but they do want to. They want to make this a useful psychological database. So we're going to get it and work on it over the summer. But
Great,
that's
thanks.
not Maddie's fault.
I understand. Thanks.
Any other questions for Maddie?
I think Geneva has one.
Okay, Geneva, yes. In the
Oh,
front there.
You stopped. She was yeah.
She liked that one.
Yeah.
And also, let me just say to the presenters, if it's at all possible for you to stay at the end, we can do a group picture with just you all after everybody else leaves. If your schedule doesn't permit, I understand. All right, so I think next up is Meg. Meg, are you ready?
Yep.
All right, so next we have Meg Houston, who's been working with Professor Martinez.
All right, can everyone hear me?
Yep. But do you want to do your screen?
Mm-hmm.
Yeah. Okay.
And then I'm gonna turn off my video just so that you guys can minimize the window so you can see everything on the slides.
Well, wait, Meg, can you just keep your video on for 1 second?
Yes.
Hold on, I'm trying to pin you. I just want to sorry to make everybody wait while I do this mom thing I do, but I'm just gonna take a quick photo.
Oh, great.
I know you love my photos.
You always catch me at bad times.
All right, well, you can pose right now. Nice.
Thanks.
Okay, you can lose your video now.
Okay. All right. Hi, my name is Meg Houston, and I will be talking today about my senior thesis, which observed that a ketogenic diet impairs condition-place preference for cocaine in Sprague-Dawley rats. As of 2017, around 5.5 million people ages 12 and over used cocaine. The highest percentage of new users, current users, and instances of cocaine use disorder were within the 18 to 25 age range. Across the years, cocaine use has remained relatively stable, making cocaine addiction a persistent problem that needs addressing. Cocaine use has been linked to numerous health risks, a few of which are cardiovascular disorders, cognitive impairment, and overdose. Cocaine acts by binding to and inhibiting the dopamine transporter, blocking the reuptake and resulting in prolonged dopamine signaling in the brain. Currently, there is no FDA-approved treatment for cocaine addiction, and despite a simple mechanism of action, repeated cocaine exposure results in a diverse range of neurobiological changes. Ideally, an effective treatment for cocaine addiction would address a multitude of these changes. A ketogenic diet is a high-fat, low-carbohydrate diet that has recently gained a lot of attention for its ability to treat a range of neurological and psychological disorders, most notably epilepsy. A ketogenic diet increases extracellular adenosine levels and therefore adenosine signaling. Adenosine is a neuromodulator which can influence dopaminergic signaling. In part due to their colocalization, which you can see in this diagram over here, adenosine and dopamine signaling act antagonistically, meaning that increases in adenosine signaling reduce dopamine signaling. Previous work in drug addiction has found that increases in adenosine signaling reduce the effects of cocaine. From this information, we hypothesized that ketogenic diets positively impact brain processes that are negatively impacted by drug addiction, thereby reducing the addictive properties of drugs of abuse like cocaine. Megan Lees, a previous thesis student in our lab, found that a ketogenic diet administration decreases cocaine-induced locomotor and stereotyped responses in rats. Moving forward, we wanted to assess if a ketogenic diet influences the motivational properties of cocaine. We predicted that male and female rats who are given a ketogenic diet will show diminished motivation for cocaine observed through a reduced condition-place preference. 78 male and female Sprague-Dawley rats were used for this experiment. Rats were pair housed by sex, exposed to a 12-hour light-dark cycle with lights on at 7 a.m. At 5 weeks of age, rats were randomly assigned to remain on the standard diet or switch to the ketogenic diet. And testing began when the rats were 8 to 10 weeks of age, took place between the hours of 9 a.m. and 5 p.m. In a rat, 5 weeks of age corresponds with adolescence, and at 8 to 10 weeks of age, they are young adults, which corresponds to the 18-25 age range that I mentioned earlier in humans. The standard diet was normal rodent chow that was comprised mainly of carbohydrates, whereas the ketogenic diet was comprised mainly of fat and had very little carbohydrates. To test the rewarding properties of cocaine, we used a 2-chamber condition-place preference box. When a person takes a drug of abuse, the environment that they are in gets associated with the rewarding effects of the drug. If a person is then craving the drug, they will seek out the context in which they took the drug. Similarly, if they find themselves in the same environmental context, they will also find themselves wanting the drug. The apparatus has distinct wall and floor patterns on each side so that they are distinct from one another. Rats have an innate preference in chamber side before they are exposed to the drug, which is determined by a pretest. During the pretest, the rats are allowed to run freely between the two sides, and time spent in each chamber is used to designate their initially preferred and initially non-preferred chamber. On conditioning days, the drug is paired with the initially non-preferred chamber, whereas saline is paired with the initially preferred chamber. If the environmental context is successfully associated with the rewarding effects of the drug by repeated stimulus context pairings, the rat is expected to switch preference, which is observed during a drug-free post-test, where the animal once again has free access to both sides of the apparatus. Cocaine hydrochloride was obtained from Sigma Aldrich, and it was dissolved in sterile saline to the working concentration of 10 mg per ml. On each conditioning day, rats received either an intraperitoneal injection of 10 mg per kg of cocaine or saline. On the right, there's a table of the treatment groups and sample sizes, noting which groups received cocaine and which received only saline. Behavioral testing consisted of 1 pretest day, 6 conditioning days, and a post-test day. During the 15-minute pretest, rats had free access to the apparatus, and time spent in each chamber was used to determine a rat's innate chamber preference. On days 1, 3, and 5 of conditioning, rats were injected with cocaine or saline and placed into their initially non-preferred chamber. And on days 2, 4, and 6, all rats were injected with saline and immediately placed into their initially preferred chamber. Each conditioning session lasted 30 minutes. During the 15-minute post-test, rats once again had free access to the apparatus, and time spent in each chamber was recorded. A CPP score was calculated to determine a rat's change in preference. The CPP score refers to the difference in time spent in a rat's initially non-preferred chamber from post-test to pre-test. Additionally, total distance traveled was measured for both the pre-test and the post-test to assess the rat's locomotor abilities. 24 hours after the post-test, Rats were euthanized and blood was collected and analyzed for blood glucose and ketone levels. Throughout the experiment, animal weight was monitored and weight measurements from baseline, pretest, and post-test were used to assess changes in weight across time. Data was analyzed using JASP. Descriptive statistics were analyzed for skewness and kurtosis so that further parametric statistics could be analyzed accurately. CPP score, total distance traveled, blood ketone, and blood glucose levels were analyzed using a three-way ANOVA, looking at the interaction of sex, drug, and diet, whereas weight was analyzed using a repeated measures ANOVA, looking at the interaction of time, sex, drug, and diet. As expected, standard diet animals had significantly higher CPP scores in the animals conditioned with cocaine versus with saline only. Interestingly, there was no effect in CPP scores regardless of injection in the ketogenic diet animals. This data suggests that the ketogenic diet blocks the formation of a CPP for cocaine, and potentially this could mean that the ketogenic diet rats had diminished motivation for cocaine. At both pretest and post-test, there was no effect of drug or diet on total distance traveled. This suggests that ketogenic diet animals were just as able to perform the locomotor functions of the CPP task as standard diet animals. As expected, there were no differences in weight at baseline, and ketogenic diet animals weighed significantly less than standard diet animals at pretest and post-test. Animals on both diets gained weight over time. The ketogenic diet animals just exhibited a much slower weight gain than standard diet animals, and this effect was seen in both sexes. As expected, there were no differences not surprisingly, ketogenic diet animals had higher ketone levels than standard diet rats. Additionally, ketone levels were higher in ketogenic diet rats treated with saline versus cocaine. Looking at blood glucose levels, we found that female rats on the ketogenic diet had lower blood glucose levels than females on the standard diet. However, there was no effect in males. In summary, we found that a ketogenic diet blocks the formation of a conditioned place preference for cocaine, and potentially ketogenic diet animals have a decreased motivation for cocaine. To explore this further, we want to investigate if the ketogenic diet has the potential to impair learning and memory processes involved in the conditioned place preference task. So we're investigating the effect of a ketogenic diet on natural rewards, such as sexual behavior. We also want to investigate the effects of a ketogenic diet on dopamine release using D-Light. D-Light is a biosensor that can be used to monitor dopamine release. And we'd use D-Light to monitor dopamine release between the ketogenic and standard diet animals to see if there are any alterations in dopamine release between the diet conditions. I would like to thank Professor Martinez, Kira, Nina, Sam, Eve, Jolie, and Maddie for their help on this project. And I would like to thank you all for listening.
Yay.
Yay!
Well
Awesome.
done, Meg. Questions for Meg?
Meg, I have a question.
Yep. I think you're still muted.
You're muted.
Sorry, I took my finger off the space bar. In the last data slide that you showed with the blood analysis,
Yep.
and you showed.
I can pull it back up if you want.
Could you please? My memory isn't what it used to be.
This one?
Yes, that one right there. So the blood glucose, were those groups split into cocaine and saline groups, and because
Yeah, so they were initially split into cocaine and saline, but there was no drug effect, so we collapsed the graph to highlight the sex effect.
okay, great. I was just curious if cocaine had any influence on.
Yeah, no, we didn't see any effect of drug on blood glucose levels.
Other questions?
Yeah, I had a quick question. So you mentioned at the beginning you start the ketogenic diet at 5 weeks when the rats are still adolescents. Does that mean their brain is still developing? And if so, do you think if you started them on ketogenic diets after they were done developing, if they'd still have the same effect on cocaine use?
So I would expect that their brains are still developing. And I would also expect that at an older age that we would see the same effects, but we wanted to highlight the effects in the 8 to 10 week old rats to kind of correspond with the 18-25 age range of the young adults being the highest users. And then the 5 weeks kind of gives us enough time so that the rats are on the diet for 3 weeks, so they stabilize to the diet. But I think in adults, we would expect to see the same effects if the ketogenic diet is increasing those extracellular adenosine levels. It hopefully would have the same effect in a developing brain and an adult brain.
Thanks.
If I could comment on that. We, in our lab, we've seen similar effects on nociception in adolescents and full adults. So it may be that the same thing would be seen with the cocaine place preference.
Any other questions?
Awesome job, Meg.
Thank you, Professor Helt.
Awesome.
All right, I think next up is Emma. Are you ready, Emma?
Yeah.
So my daughter's Emma, so it's really hard for me not to say your name like you're 2 years old because I'm just so used to going, Emma. So, but Emma's been working with Professor Guardiola Diaz.
Okay, so my thesis was on the amino acid sensing ability by the mechanistic target of rapamycin in oligodendrocytes, and my advisor was Professor Guardiola Diaz. So Katie Lazur and I have been working in Professor Guardiola Diaz's lab since our sophomore year and have been examining several questions related to the involvement of mTOR in oligodendrocytes. Oligodendrocytes are a type of glial cell in the central nervous system that surrounds the axons of neurons, which aids in the propagation of action potentials. Oligodendrocytes can project to up to 40 neurons at a time, which makes them incredibly metabolically expensive, requiring an abundance of nutrients and amino acids. So in the center of this picture, you can see the blue oligodendrocyte projecting to multiple neurons to create the myelin sheath. So oligodendrocytes go through distinct phases during their maturation process. So they start out as a pre-O2A cell and then eventually mature into a mature oligodendrocyte. There are many intrinsic and extrinsic factors that are involved in the progression of oligodendrocytes through the oligolineage. During the later stages of development, specific antigens are expressed on the surface of the membranes that are markers of the stage of the development that they're in. This can be helpful because you can utilize this to determine the specific stage they're in based on the antigen they're expressing rather than just based off of their morphology when you're looking through a microscope using different staining techniques. Previous research in Professor Guardiola Diaz's lab demonstrated that the inhibition of mTOR by rapamycin in early stages of development, like in their progenitor phases, will not stop their progression and they'll still be able to develop to become a mature oligodendrocyte. However, if mTOR is inhibited in the later stages of development, it'll cease the progression of their cytoskeleton and reduce the expression of a protein important in the myelination process called myelin basic protein. So what is mTOR and why are we studying it? mTOR, or the mechanistic target of rapamycin, is a serine threonine kinase that belongs to the larger P3K family and is stimulated by nutrients and amino acids, but is inhibited by stress, lack of nutrients, and rapamycin. It's a complex multimer that's highly conserved through evolution and is seen to play an important role in cell growth in many different systems such as yeast, algae, plants, and mammals. However, a lot of the research to date on mTOR has been done in cancer cell lines. Specifically, a lot of the articles that I looked at were in melanoma, pancreatic beta cells, or human embryonic kidney cells. Amino acids, particularly leucine, regulate the activity of mTOR through the Raptor subunit as well as the REB-GTP binding to mTOR complex 1. The activation of mTOR indirectly through leucine promotes the translocation of mTOR to the lysosomal surface, allowing it to further activate downstream pathways involved in cell growth. In these cell lines, it's shown that the presence of leucine promotes cell growth and proliferation, while the deprivation led to apoptotic cell death due to the inhibition of mTOR complex 1. Additionally, it's noteworthy to mention that there's a link between mTOR activity and the regulatory binding protein SREBP, which is a protein that regulates the transcription of genes involved in fatty acid synthesis, which is a major component of the myelin sheath. This was studied in HEC293 cell lines, and the inhibition of mTOR complex 1 diminished the activity of this binding protein. This isn't examined in my research, but it's important to acknowledge the link between mTOR and lipogenesis. So the majority of the information that we have connecting leucine and mTOR is derived from cancer cell lines, indicating that the presence of leucine allows for cancer cells to continue to proliferate. However, since these cells act very differently from regular cells, it's difficult to extrapolate the results to oligodendrocytes. So what are the goals of this study and our questions? So does leucine serve as a proxy for nutrient availability in oligodendrocytes? And is there an induction in mTOR activity upon stimulation with leucine after a period of starvation? The goal of this was because last year, biology honors thesis student Samir Madden conducted experiments examining these same questions. However, he only withdrew leucine from the cell cultures for 4 hours prior to leucine stimulation and was unable to obtain consistent results. So at the beginning of the year, we decided to see if a longer starvation period would silence the cells down so that we could see a consistent induction in mTOR activity. So at the very beginning of the year, we did IFM staining to look at the morphology of the oligodendrocytes during different periods of starvation. So we starved them for 4 days, 3 days, 2 days, 1 day, and no days. And as you see in the figure on the right, the bottom right, you can see faintly that it's like a very mature, it's immature oligodendrocyte form, and this was still seen in the day 3 cells that were starved for approximately 24 hours. So we went forward with the experiments to do a longer starvation period. So the methods for our study, we use enriched oligodendrocyte cell cultures derived from the telencephalon of 1 to 3 day old rat pups. And upon the growth factor removal, the cells were allowed to mature for 3 to 4 days. And then they were put in varying experimental conditions with varying, we did both a 4-hour starvation and 22 hours of starvation, and we looked at the effects of different amino acids as well. And so we analyzed this data by Western blot analysis to measure the phosphorylation of the downstream target of mTOR, S6K. And we also did more IFM staining to look at the morphology of the cells. So at the very beginning, we used HEC293 cells, which are human embryonic kidney cells, and GAPDH, which is an antibody, so that we could just verify that the antibodies were working and that my technique was consistent, so that any variation in band intensity was due to differences in phosphorylation level of the downstream target of mTOR rather than inconsistencies in pipetting. And as you can see, as the protein concentration was doubled, the band intensity was approximately doubled, going from around 4,000 to 8,000 to 16,000. And these are arbitrary value, numeric numbers that are just derived from a software. So our first experimental condition was 22 hours of silencing. So after 3 days of allowing the cells to grow, we removed the media and added media that did not have leucine in it. And then after 22 hours of silencing, in one of the plates, we added rapamycin for 30 minutes and then leucine stimulation for an hour as our secondary control, because rapamycin is known to consistently inhibit mTOR activity. And then our experimental condition was a 22 hours of removal of leucine and then a 1 hour stimulation. And so as we would expect, the plates right here, the LR group, had no band of phosphorylated S6K, which just confirmed that mTOR was completely inhibited by rapamycin. So that's why we see no bar over here. And all of both of these bars were derived from doing a ratio of the with the phosphorylated over the total. And you can see that when leucine was added back into the media, that there was an increase in the level of phosphorylation of the downstream target, which is shown here. And the error bars for this were standard error bars that were calculated from the standard deviation obtained by the arbitrary band intensities from different Western blot saturations. So and then our second experimental condition was the same that Samir Madden used last year in his thesis. So we did, the cells were allowed to mature for 4 days, and then leucine was removed for 4 hours, and then we added rapamycin and then leucine, and then the experimental plate was adding leucine for an hour. And these results were consistent with the induction in mTOR activity upon leucine stimulation. As you can see from this bar, is greater than the control. And again, there was no band for the phosphorylated form, or for phosphorylated S6K when the plate was added with rapamycin. But as you can see here, the induction was not as large, which is potentially why Samir had inconsistencies in his results. We're seeing that there was better results after a longer period of starvation. So moving forward, we looked at the effect of the essential amino acid methionine as well. So we set these two plates up. We no longer did a secondary control with rapamycin because it was consistently inhibiting mTOR activity. And we only did the 22 hours of starvation rather than also plates with 4 hours of starvation, just because we were seeing better results with this. And then for our last experiment, we read a paper that was examining HEC293 cells and pairing different essential and non-essential amino acids and seeing if this would affect mTOR activity. And in this paper specifically, we thought was really interesting that when the essential amino acid leucine was paired with the non-essential amino acid alanine, there was a greater induction than if leucine was alone. So we set up plates to examine that by doing 22 hours of silencing, 22 hours of silencing plus leucine stimulation, and then 22 hours of silencing plus alanine, and then alanine and leucine is the last one. Unfortunately, the last two experiments I wasn't able to obtain results from because I wasn't able to do the Western blots. So from the data that we did collect, our conclusions were that oligodendrocytes are able to withstand a prolonged 22-hour amino acid starvation period to bring the activity down to a basal level. And that mTOR activity is completely inhibited by rapamycin, which was seen by there was no phosphorylated bands. And then leucine affects mTOR activity in oligodendrocytes, indicating that mTOR senses extracellular leucine availability. So then future directions would be IFM staining. We would want to count the number of progenitors versus immature versus mature oligodendrocytes after 22-hour starvation versus after the 4-hour starvation periods based on their morphology to just confirm that there were equal number of cells in those cultures and we weren't able to do a cell count there. And also using phalloidian stains to look at the oligodendrocytes based on their antigen markers as well. And then do the analyses through Western blot for the methionine and the alanine experiments. So I would just like to thank Professor Guardiola-Diaz and Katie Lazur for helping me for the past 3 years in lab.
Good job, Emma.
Wonderful, Emma. Any questions?
Yeah, I have a question for you, Emma.
Mm-hmm.
The D3 experiment where you show that the phosphorylation of mTOR was down, or the activity of mTOR was down under, so C is the starvation condition, correct?
Yeah.
All right, so when you say 1 hour of leucine stimulation, are you just returning the leucine level in the media to sort of what is the standard level?
Yeah.
Increasing it? That's one of the questions.
We're just adding the leucine back to the standard conditions.
Okay, so that was just, that's the growth media that they spent the last 4 days in, essentially, right?
Yeah, so the media that we have in lab, we add leucine and methionine in it because it regularly doesn't have it in it. So then the media that we used during the starvation conditions just didn't have the leucine in it.
Oh, okay, all right. And then, so then the other question is, did you do any Western blots to look at sort of baseline phosphorylation activity of mTOR, just in the presence of, of growing before you starved them? Does that make any sense? In other words, when you return leucine to the media, is that phosphorylation ratio, is that just bringing itself back to normal, and that the starvation or the lack of leucine was turning off mTOR activity, or is the presence of leucine turning on mTOR activity?
Oh, I see your question. We didn't do a Western blot that.
Okay.
Right.
It's clearly sensitive to leucine. It's just a question of.
Yeah, if it just brings it back up to its normal level, or if it's enhanced. Yeah.
Yeah. Okay.
I'm not quite sure.
The defined media contains insulin, and that signals through the tyrosine kinase receptor activating mTOR. So that's really a difficult experiment to sort out.
Mm-hmm. Yeah.
And that's why starvation focuses it on in the absence of leucine and in the absence of insulin, which is something that we didn't mention.
Gotcha.
focuses the signaling on leucine.
Okay, great. Oh, and one last comment, Emma. I can't believe you couldn't convince your parents to let you set up a Western blot system in your kitchen.
I know. It's really simple.
Anyway, nice work.
Thank you.
I have a question
Awesome.
about the composition of the cultures. So what are the proportion of different types of cells that are present in your oligocultures?
So we allow them to grow on an astrocyte bed. And then after that, we're shaking the cells to remove the oligodendrocytes from the astrocyte bed. And then that's when we're withdrawing the growth factors and allowing them to differentiate. Does that answer your question?
Sure, but like if you were to do immunofluorescence for markers of oligos, you would expect to see that they're pretty much all oligodendrocytes in your culture?
Yeah.
Okay. Thank you.
Yeah, thank you.
Any other questions? All right, thank you very much, Emma. And our next speaker is Katie. Are you ready, Katie?
Yes.
All right, so Katie is our next speaker who has also been working with Professor Guardiola-Diaz.
Okay, so my thesis was focused on looking at mTOR and TRIB3 expression in oligodendrocyte lineage cells. So similar to some of the background information Emma gave, oligodendrocytes are a type of glial cell that are found in the central nervous system, and they're responsible for producing the myelin sheath, which allows for neuronal communication to be sped up. And these cells follow a very specific maturation process, which is pictured to the right over here. So they progress from early progenitors to late progenitors, then to immature oligodendrocytes before they finally become mature oligodendrocytes and are able to produce the myelin sheath. And at that mature oligodendrocyte stage, they have the many projections that are necessary to be able to make contact with the axons so that they can myelinate them. And being able to progress throughout the whole lineage and become a mature oligodendrocyte and then eventually produce the myelin sheath are very metabolically expensive processes. So it's very crucial that these oligodendrocytes are aware of the nutrients that are in their environment. So the mammalian target of rapamycin, or mTOR, is a protein serine threonine kinase that's part of the PI3K related kinase family. And mTOR has been implicated in various metabolic processes such as protein synthesis, autophagy, cell growth, and the integration of nutrient status in the surrounding environment of a cell. And as shown in the figure below, when mTOR is inhibited by rapamycin at later stages of the lineage, the oligodendrocyte is not able to terminally differentiate. However, when it's inhibited at the earlier stages, so the progenitor stages, it's still able to fully differentiate into a mature oligodendrocyte and eventually myelinate the axons. So inhibition of mTOR by rapamycin also greatly downregulates myelin basic protein, and the essential role of the mature oligodendrocytes is to be able to produce that myelin. So without as much of that myelin basic protein, it could be more difficult for oligodendrocytes to myelinate the axons. And since they're not able to fully mature when mTOR is inhibited at those later stages, there's some crucial role being played by mTOR in order for the oligodendrocytes to fully mature. So in addition to mTOR, I also looked at TRIB3, which is Tribbles3, and it's a pseudokinase that could also play a signaling role in oligodendrocytes. And it is an ortholog of Drosophila tribbles, and it's been shown to respond to cellular stress such as starvation. And as shown in the figure to the left, TRIB3 plays a role in the AKT pathway activity, which is important because AKT is also involved in the mTOR pathway. So there's the potential for the overlap of signaling between mTOR and TRIB3. And there's also the potential of TRIB3 to play a role in myelin pathology. TRIB3 expression has been increased in demyelination rats preceding the prominent myelin breakdown. And TRIB3 expression is also stimulated in the dentae gyrus by cuprizone, which is a demyelinating agent. and exact mechanisms aren't known, but research does show that there could be a possible role in this myelin pathology played by TRP3, which is obviously important since the oligodendrocytes are responsible for producing the myelin, which is why we're interested in looking at TRP3 specifically in the oligodendrocyte cells. So leucine is a nonpolar and branched chain essential amino acid, and it's been shown to directly stimulate mTOR, and we are looking at it as serving as a proxy for availability of amino acids, which is very important in the oligodendrocytes since they have to go through their whole lineage to mature, which is very metabolically expensive. And then once they are mature and able to produce myelin, that's also very metabolically expensive. It's important for these cells to know what nutrients are in their surrounding environment and knowing if they have enough to go through these processes. So we wanted to specifically look at leucine. So the two main goals of my research was to analyze gene expression in oligodendrocytes as a function of age and as a function of nutrient deprivation. And in particular, I looked at how mTOR expression is affected as the oligodendrocytes age from progenitors to mature oligodendrocytes. And I looked at how leucine deprivation affects TRP3 expression. So the first part of my methods was that I used an enriched oligodendrocyte cell culture that was prepared from the forebrain of neonatal rat pups. And I used a trizol RNase hybrid extraction to harvest the RNA. And in order to test the expression as oligodendrocytes developed, I harvested RNA at two different time points, so day 1 and day 4. So day 1 cells were more the earlier progenitor stage, and then the day 4 was closer to the mature oligodendrocyte stage. And this was to get cells at two different time points in the lineage to see how that affected the mTOR expression. And for this part of the experiment, the cells were provided all nutrients that were necessary for development using an N2 media. And then in order to test how deprivation of leucine affects gene expression, other cell samples were deprived of leucine using an N2 limb media. And I only used day 4 cells for this part of the experiment, so a matter of age wasn't affecting the experiment. And I used 2 different starvation periods, so a 2-hour, and then more recently this semester, I tried a 24-hour starvation period to see how those would affect TRP3 expression. So in order to have the quantitative data, I used qPCR to measure the gene expression, and I used the RNA that I harvested. So I used an ITAC CyberGreen one-step kit, and mTOR and TRP3 were my genes of interest, and then my reference genes were HPRT1 and GAPDH, and these reference genes functioned as internal controls, and they're not regulated developmentally or by nutrient starvation, so their expression wouldn't change under the experimental conditions I was using. And I used verified primer PCR CyberGreen assays for the primers for all the genes, and for each experiment, I used 100 nanograms of RNA from the RNA samples that I harvested in each condition. So in order to analyze this data, I used the delta-delta CQ method. So at the end of the experiment, the qPCR machine gives you CQ values for each well on the plate, and as shown in the picture to the right, so over here, the CQ value is basically the number of cycles required for the fluorescent signal to cross the threshold set by the machine. So the fluorescent dye is what allows the machine to detect the presence of the targets during each cycle, and it makes this a quantitative value. And the lower the CQ value is, that means that there were more copies of the target initially, and the fluorescence was detected at an earlier cycle. And the delta-delta CQ method is a mathematical technique that allows for the CQ values to be taken to determine the fluctuations in expression of the genes of interest relative to the reference genes. So I was looking at mTOR and TRP3 relative to HPRT1 and GAPDH. And over here, it's the mathematical equations that are used to go through each step, and I'm going to explain them a little more. So this is an example of my data. So the delta-delta CQ method directly uses the CQ information that's taken from the qPCR system to calculate relative gene expression in target and reference samples. So this is an example where I was looking at how mTOR expression was affected throughout the oligodendrocyte lineage. And in the CQ column, you can see that these are the CQ values that the machine directly gives to you. And then first the delta CQ, so in the second column, is the value that is calculated, and that's the difference in threshold cycle between the target and reference genes within each age of the cells. So I would take the mTOR CQ value and subtract the GAP-DH value from that for the day 1 cells, and then again I would do that for the day 4 cells. And then the delta CQ, which is the next column, is calculated by subtracting the delta CQ of the reference sample from the delta CQ of the target sample. So in this case, the target sample was the day 4 N2 mTOR value. So this one, or no, this one down here. So the day 4 N2. And then the reference sample was the day 1 delta CQ value. So everything is being standardized to the day 1, since that's the control in this situation, because we're just looking at the later time point, how the mTOR expression changed. So this gives a value of 0 for the reference sample, which then has a fold expression of 1 to compare the target sample to. And then the fold expression is calculated by raising 2 to the power of the negative delta CQ value. And in this example, the fold expression is down here highlighted in yellow for mTOR at day 4. So it was 2.12. So these are my results for my experiments that looked at mTOR expression as the cells were aging. So samples 1 and 3 were from the same experiment, and then samples 5 and 7 were from the same experiment, because we could only, or the same culture. And all these cells were provided all the nutrients that they needed. So they were given the N2 media, and they weren't lacking leucine at all. And up on the top here is the GAP-DH reference gene, and then the bottom is the HPRT1 reference gene. So in 3 out of the 4 of the calculations, there was an increase seen in mTOR expression observed at day 4. And as mentioned previously, all the blue bars represent the reference sample, which in this case was day 1 cells, and it has a fold expression of 1 to then be able to compare the day 4 cells to. And when using GAP-DH as the reference in the top graph, there was an expression fold change of 1.71 and then 2.12, which shows an increase in expression. And when using HPRT1 as the reference gene, there was an expression fold change of 4.29 over here, which is an increase, and then a fold change of 0.76, which is a slight decrease. And we're not exactly sure why there's not consistency when we use HPRT1 as a control. And the significance was not tested, so we're not sure the significance of it. And although there wasn't consistency with the HPRT1 analysis, 3 out of 4 of the calculations did show that mTOR was increased at day 4. And this could be because mTOR is more critical at that point, and when it is inhibited by rapamycin in later stages, the oligodendrocytes are not able to fully mature. So then these results are from my TRIP3 experiments, where I was depriving the cells of leucine. So there were more experiments for this portion, and 3 out of the 4 of the experiments were a 2-hour starve, and 1 of them was a 24-hour starve, because in the past I had been using 2-hour starves, and then this semester we decided when Emma started using longer starvations that we were going to try that with this too. So I was able to run 1 experiment with a 24-hour starve before we had to leave campus. So in the top again is GAP-DH as the reference gene, and HPRT1 down here as the reference gene. And this the blue bars were the N2 media, so these were the ones that were standardized to 1 because they were given all the nutrients. And then the yellow bars show the fold change when they're deprived of leucine. And again, all of these cells were day 4, so there's no question of age here. And the expression fold change for GAP-DH, all of them showed a decrease in expression when they were starved of leucine. So that's seen again in the yellow bars. So there was a point compared to 1, this one was 0.55, and then 0.34, 0.43, and 0.36. So that shows a slight decrease in expression. And then when we used HPRT1 as the reference gene, there was again some inconsistency with these results compared to the GAP-DH results. And 3 out of 4 of the values showed a decrease, but there was 1 that showed a slight increase. So this slight increase value was 1.11, and then this was 0.56, 0.67, and 0.36. So However, the significance again wasn't able to be determined, so we're not really sure how significant these results are, but again, there was a general trend seen for majority of the data that when the cells were starved of leucine for both a 24-hour starve and a 2-hour starve, there's a decrease in TRP3 expression. And this is also very preliminary data because we didn't get a chance to run more experiments analyzing TRP3 or adjusting the starvation period. So overall, I found that mTOR expression increased as the cells aged, and the data, again, when HBRT1 was used as a reference gene, there was some inconsistency. However, we were unable to determine why that was with the data available. And since mTOR is a crucial part of allowing the oligodendrocytes to mature, especially at those later stages, that's possibly why at that time point there would be more mTOR expression. And then the general trend seen with the TRP3 expression was that it was decreased when the cells were deprived of leucine. And this contradicts a lot of previous research stating that TRP3 expression is usually induced by glucose or amino acid starvation. And while more research definitely needs to be done, specifically in oligodendrocytes and TRP3, the reason we may have seen a decrease could be because of the starvation duration and the fact that much of this research has been done in cancer cells or cell lines, which is different than the oligodendrocyte cultures that we use. And we also don't know the significance of the data, and it still is very preliminary. So in future experiments, I would definitely want to look at TRP3 more and play around with the starvation periods and run more experiments in general. And overall, both with the mTOR and TRP3 data, run more experiments to be able to test the reproducibility of these results. And I just want to say thank you to my advisor, Professor Guardiola Diaz and Emma England. Working in lab together has been a good time. And thank you to the neuro department.
Thanks, Katie.
Great, Katie. Any questions for Katie? No questions? All right. I just want to point out quickly that we've also been joined by Professor Serafin. So I just want to sort of introduce her to everybody. Or wait, is she still there?
Yes.
Hi.
Yeah, she's live.
Hi, everybody.
Hi, Sally. Hi, Sally.
Sally and Mike have probably not met, but they're both on here. So,
Okay.
so just everybody give a warm welcome to Sally and Mike for 1 minute, and we look forward to seeing them in person on campus someday.
Absolutely.
Hi.
Awesome.
All right. So I think our next speaker is Izzy Sturdivant. Are you ready, Izzy?
Hi, yes.
Okay. So
Share my screen.
Isabella Sturdivant has been working with Professors Messino and Ruskin.
Okay. Just put that there. All right, so my thesis studied the effects of a ketogenic diet on inflammatory pain in mice. So around 20% of US adults suffer from chronic pain and 8% have high impact chronic pain, limiting major life activities. Many different types of pain disorders and pain progression disorders are due to chronic inflammation, so dietary strategies could be an effective method to manage pain symptoms and inflammation. A ketogenic diet is known to enhance adenosine signaling, which could have analgesic and anti-inflammatory effects. So the ketogenic diet is a high-fat, low carbohydrate, moderate protein diet characterized by moderate hypoglycemia, reduced neuronal excitation, and increased adenosine. In my lab, we use the BioServe diet, which has a ratio of fat to protein and carbohydrates of 6.6 to 1. Overall, the ketogenic diet replaces our body fuel from metabolizing glucose to ketone bodies. It has been found that ketones can make ATP more efficiently than glucose, and these increased levels of ATP leads to an increase in extracellular adenosine, which is suggested to decrease inflammation and pain. So here are 4 proposed mechanisms on how the ketogenic diet reduces pain and inflammation. So it either reduces excitatory and/or increases inhibitory mechanism, leads to fewer reactive oxygen species, which has anti-inflammatory effects, leads to an increase in extracellular adenosine, which has anti-inflammatory effects, or reducing glycolytic metabolism alone is analgesic. So adenosine is an inhibitory neuromodulator, and the adenosine A1 receptor is the most abundant adenosine receptor in the brain. A1 receptors are coupled to potassium channel activation and the inhibition of calcium channels, which inhibit neuronal activity and excitability. An increase in adenosine from the ketogenic diet is suggested to decrease inflammation and pain, and A1 receptor knockout mice have less adenosine signaling, which is hypothesized to cause a decrease in neuronal inhibition and anti-inflammatory effects, thus increasing excitation and inflammation. So male mice were fed a standard rodent chow diet until they were 5 to 6 weeks of age, and 4 to 5 mice were housed together in cages with ad libitum access to food and water. We used 4 different treatment groups of mice, including the wild-type mice on a controlled diet, wild-type mice on a ketogenic diet, A1 receptor knockout on a controlled diet, and A1 receptor knockout mice on a ketogenic diet. Mice were randomly selected to be on the ketogenic diet, were fed the BioServe diet, and fed this for 3 weeks prior to experimentation. So we used a complete Froun's adjuvant, the CFA agent, which is a heat-killed tuberculosis bacteria and causes persistent inflammation apparent 4 hours after the injection. We then used a tactile sensitivity behavioral test using the electronic Plantar-Von Frey probe, which tests tactile sensitivity and nociceptive pain before and after the injection of CFA. The Plantar-Von Frey was recorded 4 hours, 2 days, 4 days, and 7 days post-injection. We also measured their body weights and blood chemistry levels of glucose and ketone bodies, specifically the beta-hydroxybutyrate, and these were taken at euthanization. Our data was analyzed using 3-factor ANOVA, followed by multiple one-way ANOVAs and post-hoc tests, and data was considered significant when P was less than 0.05. So a little bit more about the Plantar-Von Frey. It measures maximum applied pressure of the hind paw until withdrawal or raising of the paw. The probe is applied to the center of the 4 walking pads on the hind paw, as shown in figure B. And the right and hind paws were tested in each mouse, allowing at least 60 seconds before testing the same paw again to reduce sensitization. 3 trials per hind paw, the right and the left, were recorded and averaged. So figure A shows that the mice rested on an elevated mesh surface so that the probe can make direct contact with the paw, and they habituated for an hour prior to testing. Figure C shows the Plantar-Von Frey probe, and D shows the paw post-CFA injection. So we hypothesized that the wild-type mice on a ketogenic diet would have increased adenosine signaling from the diet and thus show an increase in anti-inflammatory effects and a lower tactile sensitivity after the CFA injection. The A1 knockout mice on the control diet should show less adenosine signaling and thus have fewer anti-inflammatory effects and a higher tactile sensitivity after CFA injection. The effects of the diet may not be present in the A1 receptor knockout mice on the ketogenic diet, since these mice have less adenosine receptors to respond to an increase in adenosine from the diet. So the results showed, so these are the physiological results. The ketogenic diet mice had significantly higher beta-hydroxybutyrate ketone levels compared to the mice on the control diet, and they had significantly lower glucose levels and lower body weights than the mice on the control diet. So this graph shows the injected paw across all treatment groups, and ANOVA on baseline scores showed no significant difference in the tactile sensitivity prior to the CFA injection. There was a significant effect for time across all groups and diet, and the effect of diet and genotype on tactile sensitivity was also statistically significant. I also want to point out that a lower amount of pressure applied suggests a higher tactile sensitivity, indicating more pain, so lower tactile threshold on this graph. This graph shows the effect of the diet on the wild-type mice, and all post-injection points were significantly different from baseline for the wild-type mice on a controlled diet. At 4 hours and 2 days post-injection, there was a significant difference from baseline for the ketogenic wild-type mice, as shown here, and there was no significant difference from baseline on days 4 and 7 post-injection for the KD wild-type mice, as shown here. The increased sensitivity due to the CFA had reversed by 4 days in the ketogenic diet wild-type mice in contrast to the CD wild-type mice, since the CD wild-type mice still had a significant difference in scores from baseline at days 4 and 7. So this graph shows the effect of the diet on the knockout mice. The CD knockout mice were still significantly different from baseline at 7 days. But an ANOVA on tactile sensitivity for the KD knockout mice was not significant. So this shows the uninjected paw, which was the left paw, and ANOVA on the uninjected paw showed no significant changes across time for all treatment groups, except for the KD knockout mice. However, the post-hocs were not significant for the KD knockout group. So looking at the physiological effects, the ketogenic diet mice had significantly higher ketone levels, indicating that they were in fact in ketosis, which is very important in this study. The glucose levels were also significantly lower in the ketogenic diet mice, which agrees with research that has found a reduction in blood glucose levels and an increase in fatty acid oxidation on the ketogenic diet. The body weights for the ketogenic diet mice were also significantly lower than the CD mice, which has been found to be a common side effect of the diet, as the ketogenic diet can cause weight loss. So this is the result, analyzing the results for the KD wild type mice. There was a significant effect towards reduced pain, which was apparent 4 days after the injection in the KD wild type mice. The applied pressure at days 4 and 7 post-injection were not significantly different from baseline, which suggests that the increased tactile sensitivity from the CFA injection had reversed in the wild type mice on the diet compared to the wild type mice on the control diet. Again, since the control diet, wild type mice still had a significant difference in scores from baseline at days 4 and 7. These results overall suggest that the ketogenic diet could help reduce inflammatory pain and possibly result in a faster recovery than the mice on a controlled diet. So for the ketogenic diet mice, the knockout mice, the ANOVA on tactile sensitivity for this group was not significant due to high variability and a low sample size, so we cannot make a definite comparison to the control diet knockout mice. Only 4 KD knockout mice were tested, so further testing with more mice would be important to include in the future to further analyze these results. Due to current events, a different person had to run the tactile sensitivity behavioral test on the KD knockout group, leading to high variability in the scores and final results. This could have affected the data, so incorporating additional animals could help reduce and eliminate this variability. However, there is a similar trend, as you can see for the ketogenic diet mice, both the knockout and wild type mice, which are the top 2 lines. And this can infer that there could be an effect of the ketogenic diet on the knockout group as there is on the wild type mice. And if the ketogenic diet was affecting the knockout mice, this could indicate that the diet is not having an effect on adenosine signaling. Since the knockout mice would not be able to respond to an increase in adenosine signaling with less A1 receptors, this suggests that the ketogenic diet could impact inflammation and pain through alternative mechanisms independent of adenosine. So for the left paw that was not injected with the CFA agent, there was no significant changes across time for all groups, except for the KD knockout mice. However, again, the post-hocs were not significant. The tactile sensitivity scores for the left paw were consistent with the baseline scores and were not affected by the CFA injection in the right paw, as expected. Again, since the ANOVA were significant for the KD knockout group, this could be due to variability in the small sample size. So future directions will focus on increasing the sample size, specifically in the KD knockout group, to further investigate whether adenosine is involved in the effects of the ketogenic diet. We also want to look at specific circulating markers on inflammation, such as pro-inflammatory cytokines. A study on the inflammatory properties of the ketogenic diet found that rats on a KD exhibited lower pro-inflammatory cytokines than control. We could also look at local signs of inflammation in the injected paw, such as white blood cells that are involved in inflammatory processes. Research on the effects of the KD on white blood cells found that epileptic patients had a significant decrease in their total white blood cells on the ketogenic diet. We also want to incorporate an additional behavioral test known as the marble bearing test to further determine the effects of the ketogenic diet on pain. Marble bearing is a normal behavior in mice, and this could be greatly affected by pain. Using this test, we can look at whether the KD can reverse the effects that pain has on bearing behaviors as an additional method to determine if the KD can alleviate pain. We could also look at other adenosine receptors, such as the A2 receptor or other aspects of adenosine signaling, since the A1 receptor is just one component of adenosine signaling in these pathways. So I would like to thank my research advisors, Susan Messino and David Reskin, for helping me with this research, as well as my research team that, since we worked really closely this summer together, Kimber, Suzanne, and Allie, and Jenny Nord for taking great care of the animals. Thank you.
Excellent. That was great. Any questions?
It looks like Sally has a question in the chat.
Yes. Can you see it, Izzy, or you want me to read it to you?
I'm opening it now. I just have to.
She says, based on your findings, would you recommend a ketogenic diet for people suffering chronic pain?
I think that it's definitely an option to try, especially if medications haven't been effective, since it's something that you can control through your diet. I mean, it is a very strict diet, but I'm always open to people trying this for any kind of neurological disorder that they have, especially if like medications and alternative therapies haven't been effective, because we can control a lot through what we eat and what we put in our body.
Yeah, just to add to that, there's been few, real solid large clinical studies, but there are many, many reports that multiple different types of pain have been alleviated by changing your diet to lower carbohydrate or ketogenic diet.
Great. Any other questions?
Yeah, Izzy, I have a question. Were you surprised to see that sticking strictly to the control diet animals, that there wasn't really an effect of the adenosine 1 receptor knockout on pain sensitivity?
Yeah, I think that's definitely an interesting finding because, I mean, the knockout should have less adenosine signaling. So in general, they should have been different, but I think just because we didn't have a chance to really increase those sample sizes, I think our findings were kind of skewed.
Sure. Can you tell me what the sample size was for the control diet knockout group?
Yeah, we only had 4 animals.
Okay. And you had 4
for both of the knockouts. Yeah.
For both of the knockout groups. Okay.
Mm-hmm.
Wait. Izzy, I don't think that's
Yes.
true. I think that for the ketogenic diet, for the control diet wild type, there was an N of 6. And for the...
Oh, sorry. Yes. Sorry.
Also,
we had 6 animals for the control diet and 4 animals for the ketogenic diet knockout.
in the knockouts.
Yeah. So there's 2 less animals for the ketogenic diet.
Okay. And those are comparable though to your, is the 6 comparable to your sample size for the wild type animals on the control diet?
Yeah, we had 6 for both the CD and KD.
Yeah, because normally we would expect a A1 knockout to have increased pain sensitivity. Is that correct?
Yeah, higher sensitivity. Yeah.
Okay. Okay.
They have less adenosine signaling. Yeah.
I mean, Louise, it was surprising. I mean, we fully expected to see, apart from any ketogenic diet effects, we expected to see higher sensitivity to pain and paw pressure in the knockouts. That would make sense based on existing literature.
Exactly. Exactly. So do you have any questions about maybe what's going on with your A1 knockouts on that, or not really sure?
Well, I hope they're really A1 knockouts.
Yeah.
Yeah.
Okay.
We have some other animals that have different adenosine manipulations of the adenosine system. So it'd be interesting to test those as, to test the adenosine hypothesis in a different way.
Great. Thank you.
Any other questions for Izzy? All right, thank you. Next up is Anna Hackett, who's been working in my lab.
All right. Share this. So, okay, so hi everyone, I'm Anna Hackett. My thesis was the assessment of Trinity College's Return to Learn post-concussion protocol. So what is a concussion? A concussion is a mild traumatic brain injury. It results from a blow to the head or a rotational force, such as whiplash. This collision of the brain and skull can lead to shearing of nerve fibers and possible contusions. And it is so common that it's likely that every physician will treat a patient with a concussion at one point. Diagnosing concussion can be a bit difficult as it can manifest in a lot of different ways, and there are not many sensitive tests for concussion. Also, societally, we think that it is more okay to underreport symptoms than we should, but currently it can be diagnosed based on at least one of many symptoms, such as headache, confusion, or nausea. The ACRM definition of mild traumatic brain injury can be seen in the blue table to the right, and they state any period of loss of consciousness, memory loss, alteration in mental state, etc. Some of the diagnostic tests that we do use are the impact test, primarily for athletes, as well as sideline evaluations. And you can also do a battery of neuropsychological testing, such as the digit symbol substitution or digit span tests. So neurochemically, concussions trigger an acute metabolic cascade in the brain, which causes a 7 to 10-day glucose metabolic depression. This is a result from the sodium-potassium pump working in overdrive to regain homeostasis, which requires elevated activity or use of ATP, which is what leads to the glucose metabolic depression. We know that deficits in attention, memory, and executive function are often correlated with this acute recovery stage, and that's why this study will be looking at those three cognitive profiles. We also know that full recovery from concussion is seen within 3 months. However, any individual who's experiencing symptoms that last longer than 3 weeks could be a candidate for what is called post-concussive symptoms. Post-concussive symptoms affect about a third of individuals who sustain a concussion. These individuals could be diagnosed by deficits in attention or memory, and as well as 3 symptoms such as fatigue, sleep disturbances, headache, etc. It's these students with post-concussive symptoms that will need the greatest support when returning to the classroom. So why should we care about the proper care of concussions, especially when we know how common they are and how frequently people fully recover from them? Well, we know that collegiate athletes sustain higher levels of concussion than high school athletes. We also know that collegiate athletes are more motivated to underreport their symptoms. This is due to factors such as greater identity relation to their sports and greater emphasis on peer acceptance and coach support. And despite knowing the greater risk that college athletes and students are at risk for, there are less established return to learn protocols in place for college students. And while concussions do not need to have lasting effects on an individual, we know that premature exercise or uncontrolled activity too soon following their concussion could impair their recovery. We also know that cells are at a high risk of long-term damage or apoptosis during this acute recovery stage, which is why proper care is so important. A return to learn protocol is the counterpart to the more established return to play protocol. It is how we help concussed individuals return to the classroom as opposed to their athletic obligations. As of now, there are no known established return to learn protocols in place for college students. Trinity is the only college that we know of that is implementing a protocol. We also know that students who receive academic adjustments without penalty for missed work are more successful and they're better able to integrate into school. We know that limited school attendance can cause anxiety and depression for students, which can make proper recovery harder to achieve. This is because the symptoms of anxiety and depression often manifest similarly to concussive symptoms, and they can exacerbate each other. There is increasing consensus for the optimal return to learn protocol. However, there's been little research done on the subject. So at Trinity, the protocol has 4 steps. It begins with no class, followed by maximum accommodations, minimal accommodations, and a full return to classes with no accommodations. Every faculty member, upon a student's diagnosis of concussion, should be given a chart of suggested accommodations. These are categorized by general, cognitive, physical, or emotional deficits seen by the student. So this can be seen on this slide here. An example of an accommodation might be providing extended time for a student to complete assignments, or developing discrete ways for students to leave the class if they need moments of rest. So this study is looking at whether individuals with a specific cognitive profile have a greater difficulty returning to the classroom. So we are looking at deficits in either attention, memory, or executive function. We also want to know if Trinity's protocol is well known by students, faculty, and coaches, and if it's effective. We're doing this by surveying students who had previously been diagnosed with concussion, as well as current faculty and coaches. These surveys were evaluating the experience that these students, faculty, and coaches had with the return to learn protocol. Students who were willing were also given a battery of cognitive tests, and these were looking at whether or not they had deficits in attention, memory, or executive function. These cognitive test results were then compared to their survey results to see if there are any specific deficits that correlated with certain experiences returning to the classroom. Faculty and coach surveys were then analyzed to assess if any improvements could be made to the protocol currently in place. The survey looked a bit different depending on which demographic you were in. A student survey had questions such as, are you confident your symptoms will not return while doing schoolwork? Or did you feel ready to return to the classroom when you did? Faculty surveys had questions such as, was it difficult working with a student with concussion? And how motivated did the student seem to do well? Coach questions included, do you think athletes might be more motivated to return to the classroom? And how much do you personally prioritize returning your athlete to the classroom as well as to their athletic obligations? The cognitive tests used in this study were the Stroop color and word test, the animal naming test, the Cantab spatial span, the Weiss digit span, Weiss 4 similarities test, and the Weiss 4 digit symbol test. Cognitive deficits can look a few different ways in the classroom. A deficit in attention might manifest as difficulty maintaining focus in class or feeling foggy or slow. A deficit in executive function could be difficulty planning and organizing work that needs to be done, or multitasking when taking notes and listening to the professor. A deficit in memory could look like difficulty retaining information and then later retrieving that information on a test. So we hypothesized that participants with a deficit in attention will have the greatest difficulty returning to the classroom. We hypothesized this because attention is the most difficult to compensate for out of the 3 cognitive profiles. And also it is a foundational process for both memory and executive function. We also postulated that most members of the Trinity College community are not familiar with the Return to Learn protocol. In the study, we had 36 students, 24 faculty members, and 20 coaches participate. And what we found is that most students were unfamiliar with the protocol. This was 61% of the students that we surveyed were not aware that there was a protocol in place at Trinity. Also, approximately half of faculty and coaches were unfamiliar with the protocol. This was 46 and 45% respectively. So it's clear that there needs to be further education on the existence and the enforcement of this protocol at Trinity College. We also found that deficits in executive function rather than attention were shown to correlate with severity of current concussive symptoms. So students who scored poorly on the animal naming test had the most severe current concussive symptoms. So this means that we should focus on accommodations for executive function when returning students to the classroom, as well as others, but put a more severe focus on this. We also found out that severity of concussive symptoms correlated with severity of depressive symptoms. And it's important to note that both concussive and depressive symptoms are linked with cognitive deficits. So these 2 diagnoses can exacerbate each other. So there needs to be greater emotional support for students that are trying to return to the classroom following a concussion, along with the academic accommodations. Some other results of note were that there were members of all 3 demographics who voiced concern for non-athlete student support. It is likely that student athletes are greater educated on the risks of concussion and are also greater supported by the more established return to play protocols that are in place across the country. So there needs to be greater education and outreach to non-athlete students who might have experienced a concussion. We also found that only 8 students scored a score of 0 on the current concussive symptom scale. However, when asked if they were experiencing lasting effects of their concussion, 22 of the 36 students reported they had no lasting symptoms. This is in congruence with previous studies that have found that students often report not having lasting effects when in actuality they do. And while we don't know why this is the case, it could be that students are unaware of their symptoms being correlated to their concussion, or it may be an issue of underreporting. Also, though we did not find any significant correlation with attention deficits and our survey results, we did find that the most common lasting effect that students reported was difficulty concentrating. So this should be taken into account when helping a student return to the classroom. Accommodations such as allowing those rest breaks might be helpful. Next, we found that most students experienced frustration while returning to the classroom. And though this frustration likely cannot be completely avoided, it is further evidence that there needs to be greater emotional support for students who are struggling to return to the classroom following a concussion. One student commented that, "My professor's help made all the difference. It could have been much harder, which is why professors need to be aware of the impact." So this is uplifting because it shows that this student's experience was not as difficult as it could have been, but it shows the importance of educating everyone on the Trinity College campus on how to support these concussed students. Finally, most students felt prepared to return to the classroom, though 13 of the 36 students reported they did not feel ready. So while this is promising and that most students are benefiting from the protocol currently in place, there is certainly room for improvement. So conclusions are that the Trinity College community needs to be better educated on the return to learn protocol in place. Also, extra care needs to be taken to account for potential rises in depressive conditions of concussed students. So along with accommodating academics, there needs to be emotional support for these students. Next, we found that accommodations for deficits in executive function should be enforced, such as rescheduling assignments with the student or giving them note-taking aids in class as they're recovering. Next, further research needs to be implemented in the effective enforcement of a collegiate return to learn protocol, as a college setting is a much more accelerated process than high schools that have longer semesters and longer time to allow their student to return. So there needs to be further research on how to make the college protocol shorter and more effective. Finally, this study was promising in that it showed the beneficial effects that a well-enforced protocol can have for students, but it also showed that there needs to be further enforcement and education of the protocol at Trinity. Any questions?
Thanks, Anna. Any questions for Anna? Oh, Sally has a question. Wonderful work, Anna. You may have said this, but were you able to control for past concussions, and were there any sex differences in return to learn?
So we didn't analyze if there were any differences between male and female participants. And we also did ask how many previous concussions a student had experienced. We were only looking at symptomology from their most current concussion, however, which had occurred at Trinity.
It's not a question, just a comment, but I just want to say, Anna, how important I think this work is. Just from my role on Academic Affairs Committee, I can see that this is, a concussion is a reason a lot of students withdraw from a course. And a lot of times the course they choose to withdraw from is because that, that professor or faculty member hasn't been willing to be as flexible about accommodations, rather than because it's the course they need the least or something like that. So I love that you're doing this work and that you're making us all aware of what an important need this is on our own campus.
Thank you for that.
I might have missed this. Oh, sorry, is somebody talking?
Sorry, I was just going to quickly say a common comment that I got from a lot of faculty members was that they wanted to have more information about their students who had been concussed. So I think it's hopeful that there's a lot of faculty members that are interested in helping their students. There just needs to be better implementation of the protocol.
I think you're right. And I think some of the real giving out the specific suggestions that you had is a great idea.
Mm-hmm.
What I was going to ask was just, would you recommend, I might have missed this, would you recommend this kind of battery of cognitive tests for all students kind of coming back from concussions and looking to go back into the academic setting? Because it seems like you have a nice kind of battery that might be sensitive.
So an issue with that could be if the student had any pre-existing, say, like attention deficit disorder or
Sure.
that might affect their results. So it could be helpful in that it won't harm a student who's trying to get a greater diagnosis. However, there are still issues with using it as a way to diagnose concussion. And also you may have, what we found is that there wasn't a lot of variability in the tests except for one of the executive functioning tests. So I don't think it would hurt, but it might not be as effective as we would hope.
Okay.
So people looking for long-term accommodations have to get a battery like that by an independent neuropsychologist, but it's so hard right after concussion, it can increase their symptoms. And so it's a great idea, but, and something we should think about at Trinity, maybe at one of these little screening batteries that they do post-concussion might be helpful, but it is complicated.
Gotcha.
Any other questions? All right, thank you, Anna.
Thank you to everyone who participated in this study and for all your support.
All right, so next up is Ross. So Ross Saka has also been working in my lab.
Can you guys see me and hear me?
Yes.
Okay, cool. All right. Can you see it?
Yep.
All right. So my thesis has investigated the effect of acute stress on time-based perspective memory. Time-based perspective memory is the ability to remember to complete a task at a specific point in time, such as remembering to move your car at 9:30 in the morning, or remembering the need to call somebody back 10 minutes in the future. Time-based perspective memory relies on spontaneous generation of associated responses, such as requiring a retrieval mode that is thought to be mediated by frontal cortices. Stress, situational stress or acute stress, includes HPA axis activation, PNS activation, or a combination of the two. It has been found to increase frontal lobe activation in EEG recordings. Additionally, greater time-based response accuracy has been noted in response to stress, and 3 histological studies have shown the presence of cortisol receptors within structures heavily implicated in perspective memory. So the stressor used in this study is the socially evaluated cold pressor test. It couples both a physical and psychosocial stress response. The psychosocial stress response comes from the participant being asked to direct their gaze into a camera that will be recording their reaction for later behavioral analysis. During this, the researcher is also pretending to take avid notes on the participant. The physical aspect requires the participant to submerge their hand into an ice-cold water bath for an undisclosed amount of time. This amount of time is 3 minutes. The physical component is thought to trigger the parasympathetic stress response, which in turn almost immediately elevates salivary alpha amylase levels. Additionally, the psychosocial component is thought to trigger the HPA axis, which in turn releases glucocorticoids and within 15 to 20 minutes post-stressor elevates salivary cortisol levels. The control in this experiment was submerging a participant's hand in lukewarm water without any of the aforementioned psychosocial components. So some electrophysiological correlates shared between time-based PM and stress. Within Kona et al., they hypothesized that a retrieval mode mechanism was displayed by an ERP that was a positive modulation of the prefrontal frontal lobes. And ERP is an event-related potential. And this was between the time frame of 130 to 180 milliseconds. And stress relating stress to frontal cortice mediated mechanisms. Within Sanger et al., they noted that there was higher frontal lobe activation for stressed participants completing a computer-generated visual discrimination task. The protocol for this study included a collection of a first saliva sample, which was the baseline cortisol level, then a battery of questionnaires, and then either the stressor or the control stressor, which was administered by a different research assistant, which took about 4 minutes to complete. And then 2 research assistants set up the EEG, taking about 15 minutes. Then there was a collection of a second saliva sample, which should have been the most elevated sample. This was taken around 15 to 20 minutes post-stressor. And then the time-based paradigm took about 27 minutes to complete, and then after which a third saliva sample was collected. This resulted in about 61 to 71 minutes per participant. The questionnaires used in the study were the Daily Stress Index, the Beck Depression Inventory, the Beck Anxiety Inventory, and the Impact of Event Scale Revised. So previous studies that investigated the effect of stress on time-based PM performance have all noted significantly enhanced time-based perspective memory performance, specifically regarding accuracy. One study that used the same stressor in this study used a different PM paradigm, and another study that used a different stressor used the same paradigm in this study. So the behavioral task used in this study was a combination of an ongoing task and a time-based task, which was created within STIM2 software and modeled after Kona et al. The ongoing task was a letter matching task in which the participants were to indicate whether the second and fourth letters matched within a 5-letter sequence. Within this, there were 350 trials lasting 4 seconds each, and the time-based task asked the participants to hit a red button after 2 or 5 minutes. And in order for their response to be regarded as accurate, they needed to hit it within 1 minute before or after the assigned time. And there were 10 time-based tasks, 6 2-minute tasks and 4 5-minute tasks. So the EEG apparatus used was a 64 electrode quick cap with a Synapse RT amplifier, all recorded on Curry 7 software. The resulting physiological data was then processed within EEG lab. Automagic Toolbox, which is an automated compilation of toolboxes within EEG lab, was used to further process samples. These electrophysiological recordings were resampled at 256 hertz with a low pass at 0.5 hertz, a high pass at 30 hertz, and a notch filter at 60 hertz. Bad channel segment detection was completed with clean raw data, and spherical reinterpolation of noisy channels was completed. Additionally, artifact removal was accomplished using MARA EOG regression, which extracted data from the ocular channels, effectively removing blinks, horizontal and lateral eye movement artifacts. All in all, we had to omit 1 subject from the stress group due to the corruption of their data. So the event-related potentials of interest come from Kona et al., in which we were looking at 180 to 800 milliseconds post-ongoing task response. Generally, we were looking for positive frontal modulation lasting through the majority of the time frame. And the grand average ERPs came from a narrowed down electrode subset of FP1, FP2, etc. And specifically, we were looking for a positive modulation between the time frame of 130 to 180 milliseconds, as previously said. And this is a P100, meaning positive at 100 milliseconds. And this is thought to reflect the retrieval mechanism of strategic monitoring. And we further hypothesized that there would be a greater frontal positive modulation in stress individuals. So our hypothesis was, in Trinity College students, those exposed to elevated cortisol levels will have enhanced time-based perspective memory performance compared to the control group. Additionally, both stress control groups will exhibit positive modulation in frontal, prefrontal areas in the ERP's post-ongoing task response. We further speculated that the stress group will exhibit a greater positive modulation compared to the control group. So data analysis for the behavioral task was completed using a 2-tailed, 2-sample equal variance homoscedastic t-test. And the electrophysiological results were analyzed within the create a study option in EEG lab using a per mutation based test. And the questionnaires were analyzed using a simple one-way ANOVA. So the questionnaire results revealed that there were no significant differences in pre-existing stress levels between groups. And here you can see my sample size, where stress were 10 participants and non-stress were 2 participants. The behavioral task results revealed that accuracy was unaffected, but the stress group's prospective memory response timing was lesser or faster than the timing observed in the non-stress group. So the electrophysiological results above, you can see the grand average ERP waveform scout maps. These ERPs were time locked to the ongoing task response. So after somebody hits that button indicating that the 2 letters, the second and fourth, are either different or the same, we are looking at the timing after that. And the takeaway from these scout maps are that there is a greater frontal activation and occipital deactivation within the non-stress group from 130 to 180 milliseconds and 400 to 800 milliseconds. Additionally, below, you can see the grand average ERP waveform for the electrode FP1, and this indicates significant differences in activation and deactivation noted by the black bars below, if you look at the arrows. And the takeaway from this is that there is greater activation at 130 and 180 milliseconds within the non-stress group. This is a greater P100, and this is basically the opposite of what we were expecting, which was very interesting to see. So to interpret these results, we went back to an article published by Plager et al. They used the same stressor used in this study and administered the FAIR cognitive task. They noted improvement in stress groups regarding inhibition, specifically saying improvements in selective attention, which is described as a filter that decides whether information is being processed or ignored. And in this present study, stress participants had significantly lower frontal activation and faster response timing to the PM task, leading us to speculate that potentially the stress group became less engaged with the ongoing task and more engaged with the PM task via enhanced selective attention. This leaves us with the question, could stress be improving the allotment of cognitive resources? So future directions for this topic would be to replicate results using the same stressor and PM measurement. No two studies use the same combination. And more research should encompass the electrophysiological correlates of prospective memory, as there are not that many. And additionally, greater sample sizes, especially for this study, would be beneficial, as I only had 10 experimental and 2 control. Additionally, processing saliva samples to get the cortisol levels to ensure effective activation of physiological stress responses is extremely important. All studies that use a stressor, whether it be the TSST or the SECPT, all ensure that cortisol levels are elevated. So for acknowledgements, I'd like to thank Dr. Raskin for all the guidance and knowledge she has shared with me. I'd also like to thank Dr. Swart for his advice on how to arrange my protocol. Additionally, I'd like to thank Elaine Wren, Bria McDonald, and Brendan Conroy for all their help with administering the SCCPT and setting up the EEG. Thank you. Any questions?
That was great, Ross. Thanks. Professor Serafin had to leave. She just says, beautiful work, Ross. Congratulations. She asked if I could send her the video so she could see the end of your talk.
Okay, great. Yeah.
Any other questions?
Yes, Ross, I have a question. This is Dr. Church.
Hi, Dr. Church.
How are you? It's good to see you again.
Good to see you.
Yeah. So the cortisol levels were not measured. Is that correct? And
yeah, due to the ongoing pandemic, the cortisol were not.
Yeah. Okay. All right. And then does the paper that you looked back at, the 2017 paper, I think was
10 more than 29 is
okay. But did they I guess it's the Pifke paper, right?
Yeah.
Did they publish cortisol levels?
Yes, they did. So
okay.
I think the 2017 article is a literature review of other studies that have investigated the effect of stress and like situational stress and post-traumatic stress on prospective memory. And all studies that use a stressor analyze cortisol levels. So that's a major stipulation to all of these results.
Gotcha. Oh, okay. Thank you very much.
Yeah.
Nice work. Cool.
Thanks.
Any other questions?
Yeah, I think I have something halfway between a question and a comment. First of all,
hi, Professor Lloyd. Welcome back.
Hi there, everyone. Good to see you all. Looking at the relationship between stress and cognition is a very timely topic. And if I caught what you're saying correctly, the stressed subjects actually responded to the perspective memory task on a shorter timeline. They were faster in their estimate of what 2 minutes or 5 minutes would be.
Yeah, and electrophysiological correlates were less.
So that's, and you mentioned that selective attention was possibly involved in this, but I just wanted to ask or point out that maybe their subjective estimate of the duration of the interval has been altered by their stress. And I just think this is intriguing because one side effect of our current crisis is everyone has this sense all the time that they don't know what day it is and so forth. There's a lot of disorientation to the timeline that seems to be going on. Possibly stress is a kind of contributor to that.
Yeah, that's very interesting. It very well could be.
Yeah, it's really interesting. Any other questions or comments?
Excellent work, Ross.
Thank you.
All right, so I think our last speaker of the day, and thank you all for being here, for all these amazing students, is Kimber, who's also been working with Professors Messino and Ruskin.
So the topic of my thesis project was evaluating the effects of a ketogenic diet on tactile sensitivity and nociception across the estrus cycle in female rats. A ketogenic diet is a high-fat, sufficient protein, extremely low carbohydrate diet. Maintenance on a ketogenic diet induces a metabolic state of ketosis in which glycolysis is minimized and ketone bodies are metabolized as the primary source of energy. A ketogenic diet has been most notably used in the treatment of pediatric and refractory epilepsy, and recent evidence suggests other possible effects, including an ability to slow neurodegeneration and decrease inflammation. A primary mechanism thought to underlie the anti-epileptic effects of a ketogenic diet are decreased neuronal excitation resulting from increased activation of adenosine A1 receptors. Minimizing glycolysis increases adenosine levels by increasing extracellular release of ATP, which is rapidly dephosphorylated into adenosine. Activity of adenosine at adenosine A1 receptors decreases neuronal excitation by inhibiting production of cyclic AMP, which is an excitatory signaling molecule. And other potential mechanisms thought to decrease neuronal excitation are decreased excitatory glutamate signaling and increased inhibitory GABA signaling. It's been proposed that the anti-epileptic mechanisms of a ketogenic diet may also have an effect on nociception. And this idea is supported by the clinical use of anticonvulsant medication to treat neuropathic pain, which suggests the involvement of similar pathways. It's also supported by evidence showing that increased activation of adenosine A1 receptors reduces tactile hypersensitivity and thermal pain sensitivity in mice, and evidence that increased activation of GABA receptors decreases nociception in rats. These findings suggest that cellular changes induced by maintenance on a ketogenic diet may have the potential for anti-nociceptive effects. Chronic pain is a condition that affects 20% of US adults and can often be disabling and detrimental to quality of life. Current treatments for chronic pain is typically centered around symptom management using pharmacological agents like NSAIDs and opioids. However, these drugs offer poor long-term efficacy due to risks of organ damage, tolerance, and dependence with prolonged use. The limitations of current treatments highlight a significant need for alternative therapies, and evidence suggests that a ketogenic diet may present a safer, more sustainable alternative treatment for chronic pain. This possibility is supported by recent studies which have found that administration of a ketogenic diet decreases antigen-induced tactile hypersensitivity and sensitivity to thermal pain in male rats. Despite promising evidence of anti-nociceptive effects in male rats, the effects of a ketogenic diet on nociception in females has yet to be investigated. The estrus cycle is a 4 to 5 day reproductive cycle in female rats that is marked by 4 stages: proestrus, estrus, medestrus, and diestrus, which are characterized by fluctuations in levels of sex hormones, including estrogen, progesterone, and luteinizing hormone. Research has demonstrated that sex hormone levels impact pain sensitivity in females, but the findings are conflicting. For example, some studies indicate that sensitivity is increased when hormone levels are the highest, while other studies indicate that high hormone levels decrease sensitivity. Evidence also suggests that sex hormone levels may impact the efficacy of pharmacological treatments for pain. For example, several studies have found that hormone depletion through ovariectomy potentiates the effects of analgesic drugs like morphine and buprenorphine, in female rats. The objectives of this study were to determine the effects of a ketogenic diet on tactile sensitivity in female rats, which was assessed using the tactile von Frey test, to determine the effects of a ketogenic diet on pain sensitivity in female rats, which was assessed using the hot plate test, and to determine whether current estrous cycle phase impacts tactile sensitivity, pain sensitivity, or the effects of a ketogenic diet on either factor. The experimental subjects were adult female rats, 4 Sprague-Dawley and 4 Long-Evans, and were pair-housed in a 12-hour light-dark cycle, with testing conducted during the light phase, and given ad libitum access to food and water. All animals were maintained on the standard control diet during the first round of testing. The animals were then switched to a strict ketogenic diet, which was maintained for 3 weeks to ensure establishment of ketosis before testing, and the ketogenic diet was then maintained during the second round of testing. Tactile sensitivity was assessed using the electronic von Frey device to measure threshold to hind paw withdrawal from pressure. Rats were confined in individual chambers on an elevated mesh screen, and gentle pressure was applied to the center of each hind paw with a pressure-sensitive handheld probe. The pressure was gradually increased until the animal withdrew the hind paw, and the maximum pressure applied to induce withdrawal was recorded. Pain sensitivity was assessed by measuring latency to hind paw withdrawal from a hot plate at 50 degrees Celsius. Hind paw withdrawal was defined as stationary lifting or licking of either hind paw, and animals were immediately removed upon display of withdrawal and latency was recorded. To prevent tissue damage, animals are removed from the hot plate after 60 seconds if withdrawal was not displayed, in which case the latency was recorded as 60 seconds. Hot plate testing was conducted following the conclusion of tactile testing on each diet, and there was no overlap between the two tests. To determine the animal's current estrous cycle stage, samples were obtained by vaginal lavage on each day of testing using a small amount of saline inserted into and withdrawn from the vagina with a glass pipette. All the samples were collected after testing to prevent handling stress from interfering with the testing results. The samples were placed on slides and observed under a light microscope while wet. An estrous stage was determined based on the ratio of cell types. Samples were then set to dry and stained with hematoxylin and shore stains, and re-examined under a light microscope. An estrous stage was confirmed based on the ratio of cell types and colors. Samples that could not be definitively staged were excluded from the data, and data from the metaestrous and diestrous stages were combined for analysis due to the similarity of the hormone levels in each stage. Results of the von Frey tactile sensitivity testing showed that ketogenic diet treatment increased overall threshold to hind paw withdrawal from pressure. As you can see on the graph on the right, withdrawal threshold was significantly higher during ketogenic diet treatment versus the control diet treatment. These results indicate that ketogenic diet treatment decreases tactile sensitivity in females. When data were analyzed by estrous stage, there were no significant effects of estrous stage on baseline tactile sensitivity within each diet. However, between diet groups, ketogenic diet treatment was shown to decrease tactile sensitivity in animals during the proestrus and the combined metaestrus-diestrous stages, with no significant effects of ketogenic diet treatment on tactile sensitivity during estrus. So as you can see on the graph, withdrawal threshold was significantly higher in the ketogenic diet group during proestrus and the combined metaestrus and diestrus, but there were no significant differences between diet groups during estrus. Results of the pain sensitivity testing showed that the ketogenic diet treatment decreased latency to hind paw withdrawal from the hot plate. So as you can see on the graph, withdrawal latency was significantly lower in the ketogenic diet versus the control diet group. These results indicate that ketogenic diet treatment increases sensitivity to thermal pain in female rats. With regards to estrus stage, there were again no significant effects of estrus stage on baseline pain sensitivity within each diet. Between diet groups, ketogenic diet treatment was shown to decrease pain sensitivity during only the combined metaestrus and diestrus stage, with no significant effects of ketogenic diet treatment on pain sensitivity during proestrus or estrus. So as you can see on the graph, latency to withdrawal was significantly lower in the ketogenic diet group during the combined metaestrus and diestrus stage, but there were no significant effects of diet during proestrus or estrus. So in summary, the results showed that ketogenic diet treatment had opposite effects on tactile sensitivity and pain sensitivity in female rats. The diet caused a decrease in tactile sensitivity, with effects present during proestrus and combined metestrus and diestrus, but an increase in pain sensitivity, with effects present only during the combined metestrus and diestrus. The increase in pain sensitivity that was shown during ketogenic diet treatment was an unexpected result. Based on previous findings that ketogenic diet treatment decreases pain sensitivity at a similar range of hot plate temperatures in male rats, we expected to find that pain sensitivity would be decreased in females as well. The increase in pain sensitivity found in the study suggests that the mechanisms of a ketogenic diet may have the potential for pro-nociceptive effects. One possible explanation for these effects would be an increase in activation of adenosine A2 receptors, resulting from increased adenosine levels during ketogenic diet treatment. The activation of adenosine A1 receptors is thought to underlie the diet's anti-epileptic effects, but activation of adenosine A2 receptors actually produces an opposite effect and increases neuronal excitability, in part by increasing the production of cyclic AMP. And in relation to pain, it's been demonstrated in male rats that intradermal administration of adenosine increases pain sensitivity through activation of A2 receptors. Another surprising aspect of the results of this study was that the differential effects of a ketogenic diet treatment on tactile sensitivity versus pain sensitivity. A possible explanation for the difference in effects could be a difference in sensory mechanisms. Sensation of non-nociceptive tactile pressure is mediated by mechanoreceptors in the skin, while thermal nociception is primarily mediated by TRPV1 receptors. So it's possible that the mechanisms of a ketogenic diet have different effects within distinct nociceptive and non-nociceptive sensory pathways. In examining the impacts of estrus stage, the results showed that the ketogenic diet's effects on both tactile and pain sensitivity were significant during combined metestrus and diestrus, but not during estrus. Metestrus and diestrus are characterized by moderate levels of both estradiol and progesterone, while estrus is characterized by low levels of estradiol and progesterone. The presence of effects at moderate hormone levels suggests that estradiol and progesterone may influence the effects of a ketogenic diet on tactile and pain sensitivity. Estradiol administration has been shown to upregulate expression of both adenosine A1 and A2 receptors, which may be one mechanism by which female sex hormones modulate the diet's effects. This study was the first to investigate the effects of a ketogenic diet on sensitivity in females, and confirmation of the unexpected findings requires further research. Continuation of this study using a larger sample size may further elucidate the findings, as the small sample size and irregular cycling of subjects in this study limited the availability of data in some estrus stages, which hindered the results. Further investigation of the effects of ketogenic diet treatment in females is necessary to confirm potential sex-based differences in the diet's effects on nociception. The use of overreactomized females in future studies may also allow for better evaluation of the impact of sex hormones due to the availability for more precise control over the timing and levels of administered hormones. I'd like to thank my advisors, Dave Ruskin and Susan Messino, and my research team for their support and guidance throughout this project, and to Jenny Nord for taking care of the animals. Any questions?
Thanks, Kim. Can you put your video on for 1 second so I can take a photo?
Yep.
Great, thanks. Any questions for Kim?
I have a question about, is there any evidence for sex differences in adenosine receptor expression, like say the A2 receptor, that might help explain potentially why females might be experiencing more pain in when they're on the ketogenic diet as compared to males?
I didn't see any differences between like the expression of different subtypes of the receptors between males and females, but I did see effects of like the different sex hormones between the sexes and how those interact with the adenosine receptors. I'm not sure in terms of baseline expression level.
Okay.
if I could follow up on that, do you know if there's any difference in A2 receptor expression between nociceptive neurons and normal mechanoreceptor neurons?
That's another good question, and I'm not sure about that. I think we don't, from what I saw of the literature, it's not totally sure what the pathway for like non-nociceptive versus nociceptive stimuli are, and while they've identified like different receptors involved in each pathway, they're not sure the entire mechanism or the differences between the mechanisms between the two pathways.
Thanks.
Any other questions? All right, well, maybe we can have just a big round of applause for all of the presenters today. And if the presenters can stay for a photo, that'd be great. Congratulations to all of you. Yeah!
Excellent.
Good job, everybody.
Good work, everyone.
Excellent.
I know these were not ideal circumstances and you all persevered. So it's very, very, very impressive work. All right, so seniors, we'll see you tonight at dinner. Otherwise, thank you, everybody.
Everybody.
Awesome.
Everybody.
Thank you.
Thank
See you tonight.
All right, so presenters stay and I'll do a little group photo. One second. Sorry, just removing people. All right. Oh, wait, how do I get me out of it? Oh, there we go. Okay. Everyone ready? I'll do a few because otherwise Meg will complain. One, two, three. I'm gonna do another one. One, two, three. Yay. All right, thank you all. See you tonight for dinner. It'll be fun.
Thank you.
Thank you.
Bye.